Tetragametic chimerism was listed as the cause of a familial identity crisis when DNA testing initially contradicted maternity, nearly stripping a woman of her biological children. The medical examiner disagreed with the initial findings and delved deeper into the genetic complexities at play. The scene was processed by first responders before forensic personnel arrived. A routine custody battle had escalated to an unexpected legal challenge when preliminary DNA tests failed to establish maternity between a mother and her offspring. Forensic specialists later discovered that this woman harbored two distinct sets of genetically unique cells, a condition known as tetragametic chimerism. One set of cells contained genetic material differing from the other, leading to misleading test results due to sampling limitations. This case underscores the fallacy of assuming DNA testing provides infallible proof in all circumstances, highlighting the critical role of comprehensive genetic analysis and expert interpretation in resolving complex familial disputes. Chapter: Case background in a small town in the northeastern United States. On March 15th of the current year, local authorities were notified by social services concerning a custody dispute between a mother and her children's father regarding biological parentage. This case underscores the fallacy of assuming DNA testing provides infallible proof in all circumstances, highlighting the critical role of comprehensive genetic analysis and expert interpretation in resolving complex familial disputes. Initial observations indicated discrepancies when standard DNA testing failed to confirm maternity. The case was escalated to forensic geneticists specializing in complex parental disputes. Genetic analysis initially revealed that the woman's buccal swab sample did not match the DNA profiles of the two children, leading to concerns over her legal and biological relationship to them. Further investigation by a multidisciplinary team, including medical examiners and genetic counselors, uncovered evidence suggesting tetragametic chimerism in the mother. This condition involves the fusion of genetically distinct zygotes during early embryonic development, resulting in an individual with two or more genetically unique cell populations within their body. The discovery was pivotal, as it explained why DNA tests from buccal swabs did not match those of her children, while other tissues would show a different genetic makeup consistent with maternity. The case highlighted the limitations and fallibility of relying solely on standard DNA testing methods in complex genetic scenarios. Chapter: Initial findings upon arrival at the residence. First responders noted an unoccupied premises with no visible signs of trauma or abnormality within the living spaces. No deceased individual was immediately apparent. However, family members reported concerns regarding recent paternity testing results, suggesting a genetic discrepancy between a mother and her biological children. This anomaly prompted further investigation by law enforcement. Initial medical assessment did not reveal any physical injuries indicative of harm to the woman in question. Scene conditions were otherwise normal for a household environment, with no signs of foul play or natural causes of death present. The primary observation was the absence of any body, as the case pertained solely to genetic identity and familial relations. Given the reported DNA test results that contradicted expected biological relationships, forensic analysts collected samples from both mother and children. These samples were submitted for reanalysis at a specialized laboratory equipped to handle complex genetic cases. This investigation aimed to clarify discrepancies and explore the possibility of rare genetic phenomena such as chimerism. Chapter: Forensic investigation. Upon initial examination, buccal swab samples were collected from the alleged mother and her children to establish genetic profiles. Saliva stains found on household items provided additional biological material for analysis. Blood samples were also obtained from both parties during a subsequent medical evaluation. The chain of custody was meticulously documented throughout all procedures. Notably, discrepancies emerged between buccal swab results and those derived from blood samples taken from the alleged mother. This anomaly prompted further investigation into cellular heterogeneity within her body. Absent were any external physical markers indicative of tetragametic chimerism. Unexpectedly, detailed genetic analysis revealed the presence of two distinct DNA profiles within maternal blood cells, one consistent with paternity exclusion and another confirming biological maternity. The initial DNA analysis was conducted using buccal swabs from both the mother and her children, employing short tandem repeat (SDR) profiling to establish genetic relatedness. However, the discrepancy prompted further investigation through whole genome sequencing (WGS). Histological examination of biopsy samples revealed two distinct cell populations within the maternal tissue, indicative of tetragametic chimerism. Microscopic analysis confirmed the presence of cells with differing karyotypes and chromosomal markers. Additional tests included single nucleotide polymorphism, SNP arrays to detect genetic mosaicism. Tissue samples from various sites, including blood, buccal mucosa, and hair follicles, were analyzed for consistency in chimeric composition. The detection threshold for identifying mixed cell populations was set at 10% minimum contribution, ensuring sensitivity for minor cell lineages. These findings elucidated the complex genetic landscape of the mother, revealing her dual genetic identity as a tetragametic chimera. The initial forensic investigation commenced with DNA testing that revealed discrepancies between the genetic profiles of the woman and her children, suggesting a lack of biological relationship. This prompted further investigation into the maternal tissue samples for inconsistencies, leading to the discovery of tetragametic chimerism within the mother's body. Dual cell populations were identified, one matching the father's genetics and another distinct from both parents, effectively masking her true genetic identity in certain tissues. The timeline established through this forensic evidence confirmed that traditional DNA testing had incorrectly indicated a non-biological relationship due to sampling error rather than actual absence of biological kinship. This sequence clarified the necessity for comprehensive genetic analysis beyond standard protocols when such anomalies are suspected, challenging the assumption of absolute reliability inherent in conventional DNA testing methodologies. The forensic analysis revealed that the discrepancy between the woman's DNA and her children's genetic profiles stemmed from her status as a tetragametic chimera. This condition resulted in two distinct cell lines within her body, one of which was sampled for initial testing, leading to inconclusive results regarding maternity. Expert testimony would affirm that while DNA analysis is highly reliable, it can yield false negatives or positives when dealing with chimerism, highlighting the necessity for comprehensive genetic assessment beyond standard protocols. The expert would assert under oath that conclusive evidence confirmed the woman's biological relationship to her children through alternative genetic markers and corroborative medical records, underscoring the interpretive nature of DNA testing in complex cases involving multiple cell populations. Expert testimony would affirm that while DNA analysis is highly reliable, it can yield false negatives or positives when dealing with chimerism, highlighting the necessity for comprehensive genetic assessment beyond standard protocols. Chapter Resolution: The legal proceedings came to a halt as forensic genetic analysis confirmed the woman's status as a tetragametic chimera. Initial DNA testing, performed on buccal swabs from her cheek and blood samples from both mother and children, had led to inconclusive results due to sampling limitations. These tissues failed to represent the full spectrum of genetically distinct cell populations within the mother's body. Further investigation employed whole genome sequencing and comprehensive tissue sampling across multiple sites, including ovarian biopsies. This approach revealed two distinct genetic profiles originating from different zygotes, one of which was present in her reproductive organs and had contributed to her children's genetic makeup. The analysis confirmed she carried both sets of DNA within herself. This evidence overturned the initial legal presumption that maternal kinship could be determined solely through standard buccal swab testing. It highlighted the fallacy of treating routine DNA tests as absolute proof, especially in complex cases involving chimerism. The court acknowledged these findings, reinstating the woman's custody and underscoring the need for more nuanced genetic analysis in disputed familial relationships. This case underscores the necessity of comprehensive genetic analysis beyond initial DNA testing, highlighting potential inaccuracies in legal and familial determinations based solely on standard protocols. It challenges the notion of DNA as an infallible gold standard, advocating for nuanced forensic methodologies to ensure accurate biological identity verification. The file remains open,